Welcome everyone who's joined us for this taster lecture. I'm Dr. Tim Fenton. I run a cancer research group in the School of Biosciences at the University of Kent. And in addition to running my research, I convene or I'm program director for one of our master's courses in biomedicine, and I teach our undergraduates, particularly in the third year. I teach courses on cancer biology and on immunology. And I'm very glad to welcome one of our third year undergraduate students, Alana Dixon, is with us this evening as well. She's very kindly offered her time to help out and answer any questions you might have about being a student at Kent. And of course, I'd be very happy to discuss the work that we're doing in our research and anything that interests you from the lecture I'm about to give. So, as I said, I generally teach on the third year courses and on the master's courses. And cancer is, as I'm sure you can imagine, quite a complicated subject. So what I've tried to do is to bring across some of the basics into this lecture. So hopefully it's of interest to you and you can follow it, but please do feel free to ask questions at the end, or indeed just feel free if you want to unmute yourself or put into the chat a question as I go along. That's absolutely fine too, and maybe Alana can help field the questions from the chat.
Yeah, that's absolutely fine.
Great. Okay, so actually I should mention that although I'm going to give you a fairly a lecture today that consists of me talking at you, in our lectures in real life, what we're typically doing nowadays is to put some presentations, make them available to students the week before their lecture, and then they can come along with questions ready to ask, and we'll engage in more of a sort of an interactive discussion about the work. So that's how we tend to work. But tonight, as I say, I'll talk to you about a particularly important concept in cancer biology and by extension to cancer treatment, and that is an introduction to oncogenes, what oncogenes are, and how we can exploit our knowledge in the development of new therapies. Typically with any course we'll have certain recommended books. So there's a textbook, The Biology of Cancer, that has a lot of the information I'll cover in that, and that's available in the library. And I also recommend a particular review article to which we have electronic access at Kent, as we do for many journal articles, called The Hallmarks of Cancer. Okay, so just before we get into the detail of it, here's an outline of what I'm going to talk to you about. I'll give you some basics on cancer, what it is, and how and why cancers develop. And then I'll talk to you about what proto-oncogenes are and how in cancer they become oncogenes, and how this can set up certain dependencies in cancer cells that we can exploit with drugs targeted against these oncogenes. And very briefly at the end, I'll give you a little flavour of some of the research that we're doing and some of the outstanding challenges that remain in cancer therapy. So just firstly, this is where we are. So over aerial view of the campus taken some years ago, but it looks very much the same still. And as I mentioned, we are in the School of Biosciences, which is across these two quite large buildings here. And my lab is just on the back of this building here. So that's where we are, just outside of Canterbury, and still a very green campus. So obviously I spent my life really since doing my undergraduate degree and PhD studying cancer biology. And why do this? Several reasons. I guess some of you may be aware that for all of us born since 1960 in the UK, there's a sort of 1 in 2 chance that we'll be diagnosed with cancer at some point in our lifetime. And this is certainly on the increase. And a good question is why? Why is there so much cancer? And the real answer to that question, and the main answer to that, is actually it's because we're tending to live longer and we're not dying of other things. So we're dying less of things like heart disease, stroke, and infectious diseases that were, responsible for a larger portion of deaths earlier in the 20th century. So in some ways it's good news, we're living longer, we're not dying of other things, but then it means that we are sadly seeing more cancer, because cancer is generally a disease associated with aging. But certainly research is really helping to at least address that mortality. So Cancer Research UK, you've got a lot of statistics on this, and they fund some of the research that we do in my lab. Back in 1970, when a lot of sort of what we might refer to as modern era of cancer research started, people diagnosed with cancer at that time, their chance of being alive 10 years after that diagnosis were roughly 1 in 4. Okay, by 2010, due to advances in cancer research, the development of new improved treatments, the odds of being alive 10 years after a cancer diagnosis in 2010 had jumped to 50%. So twice as many people surviving cancer in 2010 versus 1970. And the aim that CRUK have stated is that within 20 years from 2010, so now within the next 10 years, the hope is to get that to 3 in 4 people surviving 10 years out from a cancer diagnosis. And that's driven by cancer research. And so that's an important reason to do it. And I would also add that, you know, having been doing cancer research for 20 years or so, we learn a lot about biology doing cancer research, and that's something that really excites me as well. So as I've said, I'll give you some of the basics on cancer to get us sort of onto that level of understanding where we can look a bit more about the detail here on the oncogenes. Now, cancer is a disease of cells. So here we're looking at a nice, normal, healthy tissue. You can see the cells all regular shape. This might be, for example, the lining of your lung, the lung epithelium, or it might be the lining of the large intestine, for example. And these cells, you can see they're packed nicely together. They've grown, they've divided to a point at which they form this tissue, and then they've stopped dividing. Contrast this with a developing tumor now, where we have a different situation occurring, where we've had an overgrowth of cells that have stopped to look like the cells around them. They've stopped to look like nice, healthy epithelial cells, and they've developed into this growing tumor. And this is obviously a problem. Now, what causes cells to go from this healthy state to this cancerous state? And that's the key question that we address in our research, and that this lecture really covers. So you may be sitting there thinking that I'm doing all the work at the moment, but even though you're sat listening, not necessarily thinking you're doing very much, in fact, in the last minute, since you've been listening to me, you've made 144 million new red blood cells. You've made 35 million new gut cells and about 40,000 skin cells. So you're actually quite busy, even though you may not be aware of that. And this all involves a process of cell division. And each time a cell divides to make 2 new cells, the cell has to replicate its entire genome, so all its DNA, and share it between the 2 daughter cells. And so it's an immensely complex process, a cell division, and this needs to be very tightly regulated to ensure that cells grow to the point that is necessary, but that they don't start to overgrow the tissues as we see happens in a tumor and in cancer. So this needs to be very tightly regulated, and it's something that can go wrong. And in the course of our decades of cancer research now, it's been possible to define a number of key characteristics of cancer cells that cancer cells have really acquired, that they need to acquire to keep surviving, that are not shared by normal cells. So key hallmarks of cancer, if you like, are the fact that these cancer cells sustain their proliferative signaling. So they're receiving signals in the normal healthy tissue. Cells receive signals when it's time to grow and divide, you know, to repopulate skin, for example, as we constantly, dead skin cells are sloughing off, and we need to replace them. But in cancer cells, these signals to proliferate and to survive and to keep growing have become sort of locked in an on state, if you like. But by defining these various characteristics of cancer cells has enabled us in turn to develop strategies by which we can target cancer in quite a specific way and spare the normal healthy tissues around. And this just shows an example of many different therapeutic approaches that are now in use in the clinic for actually targeting cancer cells in a much more refined and specific way than traditional, for example, chemotherapies, which also cause problems for our healthy cells and tissues, and hence the side effects experienced by patients receiving cancer therapy. So cancer, this change in cellular behavior, this acquiring of these various hallmarks of cancer is caused fundamentally by changes to our DNA. So cancer is caused by alterations in the sequence of DNA, sometimes in the number of copies of different genes that we have, and in the structure of DNA. So we can get rearrangements, parts of chromosomes break off in cancer cells and get fused to other chromosomes. And we end up with, for example, fusion genes that have come together in cancer. So a lot of changes are seen at the DNA level in cancer. And we know that these changes that are occurring at the DNA level are really what drive the development and the progression of cancer. And indeed what sustain the cancer when it's being treated, to allow it to sometimes evolve around that treatment and to sadly recur. And these changes in the DNA, they can be mutations that affect just one DNA base, and I'll just go and show you about DNA bases in a minute, or they can be large deletions where a large part of our chromosomes are lost, for example, or as I say, rearranged. Now, this might be a bit of a refresher for those of you particularly who are doing A-level biology, but just to go through the key, as this is commonly referred to, the central dogma of molecular biology. Remember that our DNA, our blueprint for building all of our cells and our tissues, is stored in the form of DNA, these two strands of DNA that are complementary to each other. And then it's made up of these 4 letters, these 4 bases: adenine, thymine, guanine, and cytosine. Okay, and cytosines always pair with guanines, and thymines always pair with adenines. So this is the structure of the DNA, and in order for us to produce proteins, which are the things in the cells that do all the work and make a nerve cell, and a gut cell, etc., we need to go through this intermediate called messenger RNA. So the code, the DNA code, is read and transcribed into messenger RNA, so this process of transcription, and then that mRNA is then read in the form of the triplet code. So every 3 bases in the mRNA encodes for a particular amino acid. Okay, and there are 20 amino acids are the building blocks of all proteins. So the order of the amino acids in proteins dictates what that protein is, and in turn what that protein does. Okay, and that's the process of translation, which is mediated by ribosomes, these protein synthesis factories in the cell. Now, this is if we consider this as the normal gene, and of course normally proteins are, tens to hundreds to even thousands of amino acids long. We're just showing a little example here. But say, for example, we go, and this is the DNA in a skin cell, and we've gone and maybe we lie out in the sun for a few hours and we've not got any sun cream on. We've got damage happening to the DNA. So this might be UV light, for example, damaging this DNA, or it could be, for example, carcinogens from smoke, another way in which we can damage our DNA. And this can cause mutations, okay? So here we have, for example, UV light causing this C to be mutated, so it's replaced with the T, okay? So what happens if what's the consequence of that? Well, if this T, or if this C that's mutated happens to be in a region of the genome that codes for a protein, then this T will then become a U in the mRNA. Remember, T in mRNA is represented by a U, uracil. Then the key important change here is that when this C that was initially in the code is mutated to a T, and therefore instead of having a UCG in this triplet, which codes for a serine, so an amino acid called serine. Once we've changed this to a T and this in turn to a U, we then code instead for a leucine at this position. So we're changing the amino acid that we put into this protein by having this mutation here in the DNA. And this is really important because this is what happens in cancer. And by having a different amino acid at just one position in the protein, we can really affect how that protein functions. And for example, we can lock that protein into an on state, if you like. So if it's a protein that does a particular job and it's normally regulated and it can be switched on and off, just changing one amino acid, as we'll see in a minute, can really lock it into one state so it can no longer be regulated in the right way, and this can cause real problems for the cell. So maybe it's not surprising that mutations can happen because our cells are constantly being exposed to various types of DNA damage. Fortunately, we've evolved very good ways of repairing that damage. But just to give you a sort of indication of the scale of the challenge that our body has in sustaining its DNA genomes and replicating those faithfully every time we do all this cell division that I've said is going on all the time. We have, if we look at each of these letters, is a base, okay? And these bases all pair with each other in this double-stranded structure of DNA. And we have 3.2 billion of these bases in our genome. And remember that in each of our cells, we have 2 copies of our genome. We have 1 copy from mum and 1 copy from dad. Okay, so we have in total approximately 6.4 billion bases of DNA that need to be looked after and need to be replicated every time our cells divide. So this works out to be actually, it's very, very tightly packed, but in each of our cells, of course, cells are just tiny microscopic things, we've got about 2 meters of DNA if we laid that DNA all out stretched. Okay, so that means in turn that if you did actually take all the DNA in a person and you laid it out all end to end, that DNA would actually reach from here to the sun and back several times. Okay, so we've got a lot of DNA. It's very, very tightly packaged and folded up inside of us, but that is the kind of scale of what we're talking about in terms of what the body needs to protect and prevent from mutating. And so our cells are very good at protecting that DNA. And actually it turns out that each time a cell divides, we get about approximately 1 mutation per 100 billion bases. So this is why cancer is fortunately rare and why we live for many, many years, typically without developing a cancer, is that the chances of getting mutations, and particularly those mutations that would cause direct cancerous consequences, if you like, are thankfully very low. Okay, so it's important to also appreciate that cancer develops gradually, typically over many years, and it's because we need the accumulation of mutations in several key genes, typically within the cell, that are genes that are important for controlling the cellular behaviors we've discussed, so proliferation, growth, and survival of those cells before we end up with a cancer. So this, for example, is an illustration of what happens during the progression of normal, healthy epithelium, so the lining of the colon. And this is what happens gradually over several decades, really, typically in the progression to a colorectal cancer, okay, so bowel cancer. And we go through these various stages where the cells start to grow when they shouldn't be growing. But at this point, we've got what's called a benign tumor. These cells are growing too much when they shouldn't be. And we see these polyps. You can see this in a colonoscopy, for example, if you put a camera up there. But they're not actually causing any harm, and they won't necessarily go on to cause any harm at this point. The problem comes when the cells acquire further changes, which means these cells that are in the lining of the gut start to invade down through into structures they shouldn't be in. And at that point, we have a problem, and we basically have a cancer at this point, because these cells that have overgrown are now starting to cause a problem. They've completely overgrown the tissue they're in, and they're actually breaking through out of that tissue where they arose from. And at this point, they start to break off into the bloodstream. And this causes a problem in the cancer spreads. The word for that is metastasis. Cells will come from this cancer, seed off through the bloodstream and start to seed in other sites in the body. And this is what causes the big problems for the patient. But we know that from a lot of research on looking at the DNA in these different lesions and in the tumors, we have definite events, and don't worry about the terms here, but essentially we're looking at different changes that happen at the DNA level during this progression from a small hyperproliferation here in normal healthy cells through eventually to a cancer. And as I've said, that typically takes several decades. And we have these characteristic changes that often are seen in the progression of colorectal cancer. So to summarize what I've said there, cancers, also referred to as tumors, they're caused by the uncontrolled proliferation of our own cells. And our cells can start to behave in this way if they accumulate mutations, which are, as I've explained, mistakes in our genetic code, in key regulatory genes. So if the mutations, there's a lot of our DNA is basically just sort of spacer, if you like, with lots of those 3.2 billion bases are in parts of our DNA that don't actually code for proteins. So if mutations occur outside of these coding regions, it's not so important. But if we get mutations in parts of the DNA that actually code for proteins, so actually within our genes, and particularly if those mutations are occurring in key genes that regulate these kind of cellular behaviors that are changed in cancer, this is when we have a problem. And at this point, cells that have accumulated mutations in these key genes and so are behaving as cancer cells, they're said to be transformed. So that's an important concept, cellular transformation, that transformation from a normal healthy cell to a cancerous cell, which is no longer responding to cells, to signals to stop growing, etc., etc. So now I'm going to introduce the concept of proto-oncogenes. What exactly do we mean by that term? So cancer-causing mutations affect two major classes of genes. They affect proto-oncogenes, and also on the other side, they affect genes called tumor suppressor genes. Now, today I'm just going to talk to you about the oncogenes. And so oncogenes are defined as genes whose presence can lead to cancer, and they are actually mutated versions of normal genes called proto-oncogenes that code for proteins that are involved in stimulating our cells to proliferate and survive, and a number of other characteristics. And these proto-oncogenes are very important genes. Normally, we wouldn't develop from an embryo to a fully grown person, and we wouldn't continue to grow and rejuvenate our tissues if we didn't have these proto-oncogenes that drive these kind of functions. The problem is, as I said, when the DNA encoding them gets, or when the DNA encoding these proteins becomes mutated such that it changes the proteins in a way that they get stuck in an on state, if you like, or they become hyperactive proteins. On the flip side, tumor suppressor genes, which I'm not going to talk about today, that would be the next lecture. So you'll have to come to Kent if you want to hear that one. These are genes that are normally involved in slowing down cellular proliferation, for example, and countering the activity of proto-oncogenes. So normally in a healthy tissue, these are finely balanced. The opposing activities of proto-oncogenes and tumor suppressor genes in tumors, what you see is you'll see proto-oncogenes have been activated, so they've become oncogenes, and actually we often see the complete loss of some tumor suppressor genes or their inactivation by mutation. Okay, so the tumor suppressor genes have become mutated such that the proteins they encode have stopped working, and at the same time oncogenes have become switched on, and this sets up a bad problem in the cell. So how do cells know when to proliferate? So I'm just going to look here at a couple of examples of proto-oncogenes and the kind of cellular processes that they participate in. Now, cells get signals all the time from outside of the cell. This can be from far away, for example, things like insulin that gets secreted in our pancreas and then travels and has action in our muscle and our fat far away. Or these can be very local signals that are released by cells around in the immediate environment to keep the tissues in what we call homeostasis and behaving as they should. So cells at certain points will receive signals telling them that they need to divide and proliferate and grow the tissue. And typically what would happen is these signal molecules, as indicated here in this red circle, they can't get straight into the cell, they can't cross this cell membrane. So what they need to do is they bind to receptors, so these are proteins that sit on the surface of the cell, and these proteins then relay this signal all the way through into the nucleus of the cell where the DNA is, where we can change which genes are being switched on and off, and therefore initiate a cellular response. So if the cell's being told it needs to proliferate, it's going to start switching genes on that are involved with growing and replicating its DNA and eventually the cell dividing. So what happens typically is that there are multiple proteins involved in relaying this signal from the receptor at the surface of the cell down into the nucleus, where proteins called transcription factors will change which genes are switched on and off. And often the way in which this message is passed through is through transfer of a phosphate group. So a phosphorus atom with 4 oxygens around it, a phosphate being passed from one protein effectively to another on these cascades. And I like to think of this as a bit like in a relay race when the baton's being exchanged from one runner to the next. They're passing that message on down through the cell. Okay, and the striking thing about cancer is that many of the genes that encode proteins that participate in these pathways are proto-oncogenes. So what you see typically in cancer is that either receptors or genes further down in these signaling cascades they get locked into an on state in cancer. So the cell thinks, or the cell is tricked, if you like, into behaving like it's receiving this signal telling it to grow and divide, whereas in fact the signal's not there, but one or more of these proteins has got stuck in an on position or an on state because of a mutation to the DNA encoding for it. And this is turning this signal on and making this cell carry on proliferating as if it were receiving the pro-growth signal, when in fact there is no signal there. Okay, and the proteins that mediate this phosphorylation, that pass phosphate onto downstream proteins, are called protein kinases. Okay, so a kinase is an enzyme that takes phosphate from ATP and puts it onto a target protein. So it's passing this message along. Now, a couple of examples. So you may have heard of a drug called Herceptin. Herceptin is a drug that targets one of these receptors. So in this case called HER2. And the HER2 is a protein, a receptor again for a growth factor, where in breast cancer, in about 20% of women with breast cancer, the gene encoding HER2 gets amplified. So we end up with lots more copies of the gene than we should have. We should normally only have 2 copies of each gene, one from mum, one from dad, but we can get damage to the DNA that causes errors that cause certain parts of the DNA to become amplified. And we see this in these breast cancers. We see amplification of the gene encoding HER2, which leads to an overproduction of the HER2 protein. And if there's too much of this HER2 protein sitting on the surface of the cell, it starts to basically come together and auto-activate, and this turns on signaling downstream of the receptor. And doesn't matter about the names of these pathways too much, but there are various signaling pathways that then pass this pro-growth signal initiated from the receptor down into the nucleus and cause this proliferation and survival of these cells. And this just shows a technique called fluorescence in situ hybridization, which is a way we can actually see and visualize the increased numbers of copies of the HER2 gene in a breast cancer cell. So here we see what the normal cell looks like, and each gene we should see 2 copies of. So these are probes. We've got a green probe against one gene, which is not changed in cancer, and then the red probe is picking up the HER2 gene, and we can see 2 copies of the HER2 gene and 2 copies of the control gene in the normal cell. Then we can see a cell here which has a low level amplification of the HER2 gene, so now we can see more than 2 spots, so we know we've got more than 2 copies of the gene. And here's what we see when we have a high level amplification in a breast cancer cell. We can see lots and lots of copies now of that HER2 gene, and that's then causing there to be an overproduction of the HER2 protein, and that's driving the breast cancer. But these breast cancers where we have amplification of HER2, they actually display then a dependency on that HER2 signaling for their continued survival. And this is what we mean by an oncogenic addiction or oncogene addiction. They've got so much of this signal coming from HER2 now that they become reliant on that signal. So if we use a drug then to block the signal coming from HER2, we can get a very good and dramatic therapeutic response against that tumor, because those cells then die. They're being starved of that signal that they've become to rely on totally for their continued growth and survival. So I'm just going to show you one more example of that. There are many now, and we've really taken advantage of this concept to design improved therapies for many different types of cancers, but this is quite a key one too. This concerns the activation of a particular proto-oncogene called BRAF. So BRAF sits in one of these pathways. So you can see a receptor here and the nucleus down here, and we're changing the expression of genes when we get signals coming through these pathways. And RAF sits here, okay, in a pathway called the ERK pathway, kinase pathway. And what RAF does, so it's one of these protein kinases. So when RAF gets switched on, it puts phosphates onto serine amino acids on the next kinase along in the cascade, which is called MEK. And this switches MEK on. These phosphates being added to MEK causes it to be activated itself. And it then, in turn, phosphorylates amino acids on the next kinase down, which is called ERK. And then ERK finally goes into the cell nucleus, where it phosphorylates transcription factors, which then, once they've been phosphorylated, they go and sit on parts of DNA called promoters and activate the genes downstream of those promoters. So they switch certain genes on that are involved in the growth, proliferation, and survival of cells. Now, if the gene encoding BRAF acquires a particular point mutation, which is often seen in skin cancer, so it's a mutation caused by ultraviolet light, that changes just one amino acid in this big BRAF protein at position 600. So out of, well, over 600 amino acids in BRAF, just this change of one amino acid, so normally there should be a valine there, and it gets changed to an amino acid called glutamic acid. Just this one switch changes the function of this BRAF protein such that it no longer needs to be activated by the protein upstream of it, which is called RAS. It's actually just what we call constitutively active on its own. So it keeps doing this phosphorylation of MEK. So this whole pathway gets switched on constantly in these skin cancer cells. So this was observed, and we developed, not we, I didn't do this, but a drug against BRAF, so a drug that blocks this kinase activity of BRAF was developed, and it's called vemurafenib. And what we can see here is data from a clinical trial that was one of the trials that led to the approval of vemurafenib for the treatment of advanced skin cancer. This is for metastatic melanoma, when sadly the skin cancer has spread beyond just the site on the skin where it can be removed surgically, and it can go, unfortunately, to places like the brain and the lungs, and it's a very lethal cancer at that point. So you can see this is looking at the survival of patients at this point. So they've already got a very poor prognosis because it's an advanced cancer. You can see with the traditional chemotherapy, these patients do very poorly, and that vemurafenib actually, although we're still only looking at a matter of months, it significantly extended the life of these patients with this very advanced form of skin cancer. So this is based on the fact that we have this constitutively active BRAF, and if we use the inhibitor, then we remove the phosphorylation downstream and we switch off the signaling downstream of BRAF. And like with those breast cancers that have the amplified HER2, these skin cancers with the activating mutation in BRAF, we see a very dramatic response in the cells. The cells die very quickly upon BRAF inhibition. So vemurafenib, along with some other recent therapies based on harnessing our immune system, have really caused a real revolution in the treatment of skin cancer. One key challenge, and I'm going to sort of finish up by talking about this, and it's something that several of us doing cancer research at Kent, myself, my research group, also research groups of Professor Martin Michaelis and Professor Michelle Garrett, we're really addressing the issue that cancers can become resistant to these targeted therapies. So on the one hand, these targeted therapies are very exciting. They have often many fewer side effects than we would see with traditional cancer chemotherapies, and they can produce some very stunning clinical responses. But sadly, we still need to contend with the issue that tumors can frequently and do become resistant to these therapies. So what might happen, as I show you in the example, is that we'll have a tumor that we treat with our, for example, our BRAF inhibitor, and maybe the vast majority of cells within that tumor are sensitive to that inhibitor. So over that time that we give the therapy for, we kill almost all of the cells in this tumor, and the patient experiences a remission from their cancer. But sadly, if there are the odd one or two cells in this tumor that were resistant to that inhibitor, eventually we get the regrowth of the tumor and a recurrence. And we see this problem as well with conventional chemotherapy, this problem that we get eventual recurrences of tumors. So this can either happen due to there being some existing resistant cells within the initial tumor, because we have some variety of the different mutations that are seen in these cancer cells, or we can get the acquisition of resistance during treatment. Okay, so we can get further mutations occurring in the cells that make them resistant to our treatment during that time. And that's a work that we're doing a lot of work on in my lab, looking at a process by which these mutations occur during treatment. And we use cancer cell lines and also mouse models to study that in the lab. Okay, so we can see this sadly in patients. As I've said, if we look at an example of vemurafenib, this was from, again, some of the early clinical trials with vemurafenib. This is a patient who, as you can see here, sadly has a very advanced metastatic melanoma. So these nodules under the skin are where the skin cancer is metastasized from its primary site. And it's now, as you can see, this patient has tumors, all over. Now, this patient was basically at the point where they were in a hospice. At this point, they were, had probably weeks left to live, maximum. And you can see the power of some of these therapies after they've been on vemurafenib, the BRAF inhibitor. So this patient's BRAF gene was sequenced. It was found to have that V600E mutation. They were put on vemurafenib. So this is a tablet, once a day tablet. And look, you've got a very stunning clinical response here. These tumors have basically regressed, and this patient is looking far more healthy at this point. They've been able to go home, and this is the kind of response that you can get with these therapies. But very sadly, if we look again at the same patient after 23 weeks, so only 8 weeks after this photo was taken, the tumor has come, the cancer has come back with a vengeance. And at this point, it's now resistant to the vemurafenib. So this is the key clinical challenge that we're faced with. It's how do we try to learn how to combine different inhibitors to prevent this resistance from occurring? How can we learn how to use, for example, blood samples to detect early whether resistance is occurring? And how can we develop additional therapies that we can combine with these treatments to prevent this drug resistance? And this is what we spend our time studying in the lab, as I've said, using cell models and animal models, etc., and a lot of DNA sequencing technologies. So to summarize, hopefully I've explained that proto-oncogenes are genes that are normally very useful in helping our tissues to grow and to maintain themselves, but if they happen to acquire mutations, this can cause them to, the proteins that they encode, to become constantly in an on state, and this is a bad thing. We need to be able to turn pro-growth signals on and off, and so they become oncogenes. So onco from the Latin, basically for tumor. So cancer-causing gene, essentially. If they acquire mutations that switch them on, or if they become aberrantly expressed, like that case where the HER2 gene was amplified, that would still count then as an oncogene. It's being expressed in an amount much larger than it should be. But we can take advantage of knowing about these oncogenes because we know that cancer cells can become dependent on their activity. And this means that we can use these targeted therapies. There's now over 75 of the protein kinases in our genome have had approved therapies for cancer directed against them. So this is a really rich field for the development of new medicines for cancer. And part of this, and why I mentioned that personalized or precision medicine, is that we know that these therapies will only work if the mutation is there or if the target is there. So if we have someone with a skin cancer without a mutation in BRAF, they won't get a clinical benefit from having a BRAF inhibitor. So along with the treatment, we also have the diagnostic test. In this case, they would sequence the BRAF gene in the skin cancer cells and see whether it's mutated. Or in the case of a woman with diagnosis of breast cancer, one of the first things that would happen would be a test to see whether she had a HER2 amplification in her breast cancer. And if so, then she would receive the Herceptin treatment. But if not, we know that that's not going to do any good. So we'll give a different treatment. So it's this thing of understanding the changes that are happening within the cells. So understanding which oncogenes are being switched on then guides how that patient is then treated. So that really shows the real fundamental importance of understanding the biology and the genomics of these cancers. And just finally to say that resistance remains a key challenge. These tumors can initially respond to these treatments, but then sadly we often see recurrences, and that's where a lot of the work is ongoing at the moment, and particularly here at Kent, this is a big focus for those of us working on cancer research here. So I think I'll stop there. Very happy to take any questions. Also, as I said, Alana was happy to sort of talk to you about her experiences doing biology degree here at Kent. Normally we would, as I say, at the end of the lecture, have some questions, maybe now doing some little multiple choice questions to just ensure that you've understood what we've heard in the lecture, and then obviously on to the next week. So thank you.
Yeah, so we already have a question in, if you'd like me to read it out.
Great, yeah.
Yeah, so the first question we've got is, recent data released in several countries showed that in 2019, the average life expectancy of cancer patients has improved only by 1 month compared to 2009. Is it reasonable to think we can improve much more in the next 2 decades so that we will reach Cancer Research UK's goal of having 75% of patients living at least 10 years?
I think it's an ambitious goal. the other thing that's important to bear in mind is that, and you'll have heard as I've gone along, I've spoken about very specific cancers. So I spent quite a bit of time talking about skin cancer. I mentioned breast cancer with regard to Herceptin. So different cancer types have very different prognosis, and different cancer types have had greater or lesser success with the development of improved therapies. So there are still cancer types such as pancreatic cancer where we haven't been able to really do anything to improve survival. Another one would be glioblastoma multiforme, the most common brain tumor in adults. I spent a lot of time working on that in my postdoc in California. That one, we've seen very little, sadly, improvement in overall survival of patients. They still have a median survival time of approximately 14 months post diagnosis, and that's really not improved much in the last 20, 30 years. So certain cancer types we've had very little progress still because we still don't understand enough about them. Certain cancer types we've had rather dramatic progress. And I would say, I didn't speak about immunotherapies today. I've spent quite a bit of time teaching on that in the third year courses that Alana's been doing. But we have real game changers in, for example, skin cancer. So along with vemurafenib, which I showed you, which has had real benefits for patients, some of the immunotherapies have turned metastatic melanoma, so metastatic skin cancer, for those patients who get a clinical response to immunotherapy, which now is about half, we have a very, very high chance of being alive still 5-10 years out from that diagnosis, which is very different to the sort of weeks to months that would have otherwise been the case. So in certain cancers, and lung cancers are another very, very lethal cancer, responsible for the highest burden of death due to cancer worldwide, lung cancer. That's another one where we're seeing very dramatic improvements with immunotherapy and also with targeted therapy in lung cancer in recent years. So I think it's cancer type by cancer type, and it depends how you measure it. So if you average it out across all cancers, yes, you're going to see a pretty incremental improvement in survival for patients. The other thing is of course, we have health disparities, and this is a huge issue in cancer, both in cancer prevention and in cancer treatment. We in higher income countries are lucky to be able to afford some of these very expensive treatments, and we have expensive or relatively expensive screening services. In the case of certain cancers, for example, caused by HPV, we have HPV vaccination, which will prevent things like cervical cancer. Of course, in lower and middle income countries, the situation is very different. So there's much less screening. It's much more difficult to get access to these, for example, HPV vaccination. And then when we actually get to the point of a cancer diagnosis, sadly, there's much less that can be done for patients. So yes, when we look across on a worldwide level, there is a huge challenge in translating some of these developments and the developments of new therapies out to make sure that people who need them can get access to them worldwide. Yeah, but in the UK at least, some of these targets are, I think, they are feasible, admittedly challenging, but feasible. And of course, prevention is always better than cure. So there's a lot around actually earlier diagnosis, earlier detection of cancer, and indeed prevention by lifestyle changes. Smoking cessation had a huge impact on cutting rates of lung cancer and head and neck cancers, for example.
Okay. Perfect. We have a second question as well. So are there any fears that cancer research could lead to exploitation, for example, reversing aging, since some hypothesize aging is merely a natural form of cancer?
So I would say aging, cancer is one consequence of aging. So yes, sadly, there are occasional, very rare, fortunately, childhood cancers, and we all know about that. And one major goal, obviously, of cancer research is to find better treatments for those. But typically, our major risk factor for developing cancer is age, because the statistical chances of acquiring mutations in these proto-oncogenes and tumor suppressor genes increases with time. Now, treating the cancers themselves is not the same as trying to, for example, reverse the aging process. If we were to modulate aging via, again, one way you could argue in which you can effectively delay the aging process is through lifestyle changes. We know that there's a difference between biological age and chronological age if we look at things like heart health. So some of these interventions we can take to limit our cancer risk will also potentially effectively prolong our healthy lifespan. Now, in terms of the ethics or otherwise of looking at interventions that have an impact on aging, that's a slightly different field. But yes, we would potentially expect things that have an impact on aging to possibly have a protective effect against cancer development. But I don't think we should confuse treating a cancer with somehow, if you like, for lack of a better expression, playing God in terms of interfering with the natural aging process, would be my take on it. But you could have a good discussion about that for, a good few hours, I would suggest.
Yeah, it's very ethical in that sense of reversing aging, which again is something we do cover as well, when some people even do their final year projects purely into ethics and whether things like this would be okay to do and whether it's feasible as well. So
yeah, it's a very good point, Alana. I think one thing that you'll learn over a degree is you'll become a scientist, and whether you continue to be a scientist and go into research or whatever you go and do, you'll have a greater appreciation for what science is all about. And a big part of that is the ethics of doing things. Just because we can do something, should we be doing it? And that's a really important, and it comes into a lot of areas of research. Also, the whole area of we use mice to an extent in some of our research, because it's been judged that the benefits outweigh the harms there. But that's something that again we very carefully scrutinise the ethics behind everything we do before doing it.
Yeah, exactly. And I think a lot of people don't realise of how controlled that process is as well. Like, there's so many laws behind it. There's like officers that are on each university that monitor that to make sure that we're not exploiting the use of animals on campus. Yeah. But that's actually something I think we learn in second year as well. That's in one of our modules. We have a whole lecture based purely on the ethics of the use of animals. And then different ethical considerations that we have to make if we're considering proposals. And that's, it's something you learn.
Yeah, I think in the UK that is possibly the country in which we have the strictest rules on that, and it really is very, very well regulated, and rightly so.
Mm-hmm. Absolutely. I'll do that one. But I had a quick question as well. So I know obviously there's a lot of things in the press at the moment about obviously the COVID vaccine that's coming out, and that's been done so quickly. So why isn't it that we've got a vaccine against COVID so quickly, but not necessarily against cancers? Because I know we have things like HPV, but why can't we have a vaccine for every type of cancer?
Yeah, it's a good question, Alana. I think yes, the development of COVID vaccines, which is fantastic, has shown how quickly science can work, given the resource. COVID is one virus, and so the challenge in each individual patient is a very similar one. We need to prevent the virus from either infecting or causing disease in a person, but we're always dealing with the same virus, at least in the absence of mutations in the virus that would change it in a meaningful way. In cancer, the problem we have is that each cancer is different. And this is why personalized medicine becomes a very important concept, because no two cancers are the same. Therefore, we can't often have we can't have, in a way that we can vaccinate against a virus, we can't vaccinate against a tumor. We can vaccinate against certain viruses that cause tumors. So in the case of human papillomavirus, it causes cervical cancer. We can prevent those cervical cancers now by making sure people don't get HPV infection by vaccinating them, which is a great thing, fantastic outcome of cancer research, which should impact across the globe, actually, because we're starting to roll those vaccines out globally. But in terms of a cancer that's actually developed, we know our immune systems can respond to and detect developing tumors. But once a cancer has got to the point of diagnosis, it shows that our immune system has somehow been suppressed by the tumor, or the tumor has escaped that immune control. And there have been many attempts to try and generate therapeutic vaccines or generate immune response against tumors. And we're just starting to see more refined versions of those now, finally starting to have some real clinical impact, actually. But with those vaccines, they have to be tailored to the exact tumor that's developing that patient. So we use DNA sequencing to find out the changes that have occurred in that exact tumor. And then we tailor make a vaccine based on the changes we see in that tumor. So it means, of course, it's a very expensive process, but nevertheless, it's an exciting area in which we're seeing some very interesting clinical responses in clinical trials, effectively giving people immunity against their own tumor.
Yeah, so it has to be personalised. It's not going to be one that's going to be off the shelf.
Yeah.
And that you can just give to anyone. Okay. And we have another question that's just come in as well. So it says, what would you say is the largest barrier to finding more information on cancer, apart from the fact that, apart from the vast range of cancers that there are?
It's a great question. I think one of the key barriers that did exist that we've really sort of smashed through now was the cost of DNA sequencing. And that has tumbled dramatically in the last 20 years since the Human Genome Project throughout the 90s sequenced the first human genome. Took about 10 years and cost about forget the exact figure, around the order of $3 billion. We're now able to sequence a genome in an afternoon for not much more than about $1,000. So that has had an enormous effect on our ability to understand cancer in the last 10 years, really. And a lot of our research is based on breakthroughs that have come as a result of being able to do whole genome sequencing on cancers. I think in terms of further obstacles, a big barrier is the heterogeneity that exists with any one tumor. So not all tumor cells within the tumor are identical, because this tumor has been developing over years and mutations are occurring in different cells. We have, if you like, a population there of cells that has a variety inherent within it. And although we can sequence and find mutations that are present in lots of the tumor cells, it's very difficult to find those very rare mutations that might be the ones that are important in driving resistance to our therapies. So, if we have a tumor with 100 billion cells there, then it only takes a few of those cells to be slightly different or to have acquired an additional mutation for them potentially to be resistant to our therapy that we're giving to the patient on the basis of what we see as being the mutations present in the majority of their cells. So this real fundamental challenge that a tumor, it can be seen as like a population. It's a bit like thinking about antibacterial resistance. If we use an antibiotic too much, then there will be some bacteria that become resistant to antibiotic or are resistant, and they will start to outgrow. And this is the problem we have with antibiotic resistance. The same way with cancer, if we're applying a really toxic therapy or a therapy that's very efficient at killing the cancer cells, but there are one or two of those cancer cells that are resistant, then those will eventually grow through. So we have this strong evolutionary process going on within us when we're treating a cancer, and we're applying a strong selective pressure, but that enables, if the cancer's got variety within it, then it can adapt and overcome and evolve. And I think that evolvability of cancer, it's even faster, you know, the fact that it has an unstable genome, you know, there's mutations occurring because cancer cells sort of lose their ability to repair their DNA as they should, gives it an even faster evolvability. So we see that evolution happening before our eyes every time we treat a cancer patient. That's a key challenge to try and overcome.
Okay, and that's funny you mentioned that as well, because I've literally just had a lecture on it about 3 hours ago about the heterogeneity of tumours and that. So it is heavily focused in final year, but we do learn about it earlier on. But I think I'm taking at least 2 modules this term that are purely cancer based.
So you're doing biomedicine, I'm guessing, Ilana. I should know that.
Yeah, I am. So this term I'm taking cancer biology, frontiers in oncology, and then there's advanced immunology, where the second half is taught by Dr. Fenton as well, which is quite heavily based on cancer and immunotherapies and stuff. So about 2.5 of my modules are cancer based this term. Yeah, but they're really interesting and all very, very different as well. So you learn a lot of different aspects of cancer. It's not just a repeat of the exact same stuff over and over again. So we have 2 questions actually. So the first one is, what's your views on different natural treatments to cancer, such as wasp venom?
I think it's something where we have to be very careful what we're talking about when we talk about natural. So natural products have given rise to some of the most potent anti-cancer drugs. So taxanes, so there's paclitaxel and related drugs are used very widely in cancer chemotherapy, breast cancer treatment, cervical cancer, many others. Taxanes are originally isolated from yew trees, so that toxic compound in yew trees. So yew trees growing in graveyards all over the country, and are meant to ward off all evil spirits. So yes, certain very potent cancer anti-cancer compounds have originally been isolated from plants. There's slightly different, but rapamycin, which is sort of analogues of it, are used in cancer therapy, and it's also used as an effective immunosuppressant for patients who've had transplants. That was originally isolated from a fungus on a place called Easter Island out in the Pacific. So Rapa Nui is the local name for that island, so rapamycin. So we've got a lot of very, very important drugs from the natural world. But these are compounds that have been isolated from these sources and then very well characterized, purified, and we know exactly what the active ingredient there is. And they've gone through, obviously, the systematic clinical trials required to prove their efficacy. The danger comes in when there are certain, if you like, there can be things that may have activity, but it's important to understand what the limitations may be of something that's not been through a clinical trial, and what the dangers are in resorting to what might be described as like alternative remedies. I used to work in very close collaboration with the head and neck surgery unit at UCL Hospital, and I used to go to their multidisciplinary meetings each week where they would discuss each patient in turn and discuss the treatment plan for them. And sadly, from time to time, because that was a big centre, then we would have patients would be referred sometimes from more local centres when they had particularly advanced cases of cancer that couldn't be dealt with locally. They'd get referred to there as a sort of, if you like, a specialty surgery centre. And what we would sometimes see would be patients who had been diagnosed with a cancer and they had decided they were going to try and treat it if you like, sort of a natural remedy, and that would invariably go horribly wrong. And sadly, they would not. It would be too late by the time they then had a huge ulcerated tumour that, needed a very sophisticated surgery to try and remove. But at that point, sadly, it was usually palliative doing so, because there wasn't really anything that could be done for them. Whereas if they'd initially, had a more conventional treatment upon diagnosis, it's very possible that their prognosis would have been a lot better. So I think there's a lot of, this is something that you learn as well doing a science degree. Hopefully you learn to sort of critically analyze the evidence behind stuff, and to see, what is a valid, for example, therapy, or what is something that's been sort of put out there, but without the necessary evidence to support it. So that's an important skill you learn as well. I'm not trying to rubbish anything that's come from the natural world. Of course, as I say, some of our most important drugs have been isolated from various different organisms, plants, fungi, what have you. But yes, the idea that you can sort of treat cancer by, drinking a certain type of milk or something, you can, if you Google it, you can see anything online, and it's trying to separate the fact from the fiction there, and just understanding that there are people trying to make money out of selling someone a lie.
Yeah, absolutely. And I think this is one of our final ones, unless any more come through. And we said, in your view, will there, and to what extent, be any positive or negative effects of the COVID-19 pandemic and Brexit on cancer research?
Yes, well, there's another one I could talk about all night. Sadly, Cancer Research UK, who fund some of the research in my lab, they've been hit badly by the pandemic, largely because they had to close all their shops. And their shops are a very important source of income. It's like British Heart Foundation have really suffered as well. So that's been a very, very sad effect for a lot of our charity sector in the UK, is that they've been really badly hit. Hopefully that will bounce back. I was talking to one of the directors of the CRUK Centre up in Manchester last week. She came and didn't come, but she gave a virtual seminar. She thinks that that people are, in this country in general, they like to give money for Cancer Research UK. It's a popular charity, just as British Heart Foundation and others are. And hopefully once shops can reopen and people can start spending money again, then that will rebound. But of course, with the unemployment rising, then one thing that suffers is charitable donations. It seems that government funding into science is strong and should remain so. Having said that, exiting Europe is inevitably will have bad effects for our universities, for scientific collaboration within Europe, and also from the fact that we actually, as a country, the UK, did very, very well indeed out of science funding from being in the EU. So we were a net gainer, if you like. We put in a lot less money than we got out each year from the EU research funding pots. We typically got between 500 million and 1 billion a year from the EU in excess of what we put in terms of research funding. So something will have to help plug that hole too, unfortunately. But hopefully some kind of common sense can come in here, and we can get some kind of a deal that allows us to continue to collaborate in the way that we've enjoyed for the past 40 years or so, and that can try to sustain the research. So it's still not quite clear which schemes we're going to remain a part of and pay into. But as I say, it's an important area to pay attention to, yeah, certainly.
Mm-hmm. And that's something I've noticed, especially being in final year, you have to read a lot more articles and scientific articles compared to the previous years, and you'll find that at the top of a lot of them it will be a couple of scientists from this particular lab and then a couple more from a lab in a different country. Like even in our university, we had a breakthrough. It was Professor Martin Michaelis, and that had a breakthrough with possibly a treatment for COVID. And that was in collaboration with a German lab, I believe. So yeah, it's kind of it's really important, and you start seeing it a lot more. And that obviously when you're reading the articles and you're getting involved. But
Yes.
did you find that there was massive problems with like social distancing in your lab as well? Have you had to reduce?
Yeah, so that's true. That's a good point on a logistical, on a purely practical level. Yeah, we're having it still at the moment. So we've got less people in at any one time than we would normally have. So people having to sort of stagger their experiments out and book into times when they can be at the benches. They're having to wear face coverings in the labs at the moment. So yeah, it's impacting, certainly, and it has impacted. And I've just heard that there's a big worldwide shortage at the moment of pipette tips, which is a big problem because we get through thousands of pipette tips doing our work. So just on a level like that, I would echo what you said about the collaborative nature of science, particularly, biomedicine, many others. Paper that we've currently got in revision was done in collaboration with surgeons and pathologists in Norway and Austria. There are, collaborate with the States a lot. It's, sort of international collaboration is key to science, and it's a fantastically fun part of being a scientist. I've been to some really places that I would probably not have been to if it hadn't been for science, in terms of conferences, South Africa, Portugal, the States many times, Croatia, all kinds of places for meetings as well, which has been great.
Yeah, because it's partly about different expertise, but then also just equipment as well. Because there are certain labs, there's only certain labs in the world that will have a piece of technology that you'll need for your research. So it's really important that you collaborate with those labs in order to get access to it as well.
In fact, one piece of equipment, which is called laser tweezers. Kent is the only place in the UK to have this piece of equipment. We just got this year for looking at sort of the single molecule interactions between DNA and proteins. Neil Cadd in the department, who's a biophysicist. Absolutely, specialist pieces of kit that are very, very expensive. That was about £700,000, I think. So you don't want every place to have to buy these really expensive pieces of kit. So you'll end up with centres that have expertise in certain things, and then you collaborate with the people with that kit and with that expertise.
Mm-hmm. And that's also something really else important that we should note as well, that especially in your final year, if you do final year lab projects, for a lot of people, they do get the opportunity to actually go up into the research labs and use that specialist equipment and that as well. So I'm not personally doing a lab project this year, but I've got a couple of friends that are. And if COVID obviously wasn't happening, they probably would be allowed into the research labs, because that is quite normal.
That's true. Sorry, I was looking off into the corner because I had my pictures of where we were talking up in the corner of my screen. I just put it back into front of the camera now. That's better. I can actually see what's happening. Yeah, so hopefully that's given you a bit of a flavour for sort of what we do, kind of stuff that you learn about, and maybe a bit of a feel for what happens in biosciences, at least here. Not here, I'm in my house at the moment, but just down the road in Canterbury. Anything else, Alana, that you'd like to add?
I would say that I've absolutely loved being at Kent. I'm so glad I chose it, and that I had so many different options when I decided that I wanted to come to university. So if you do get the opportunity, I would recommend coming down onto campus, obviously when everything is hopefully back to normal at some point. Even if you can't do like a guided tour, just come down, walk around campus and get an idea for it. Because that was something that I really loved. I personally come from somewhere that's surrounded by countryside. So to come to a campus that was also surrounded by greenery, it felt like a second home. So yeah, that's definitely something I would recommend. But if you don't get the opportunity, there's also some 360 campus tours as well. And that's definitely look at those online, because it will give you an idea of space on the campus and different accommodations and stuff. But yeah, I've absolutely loved it, and I got the opportunity to do a placement as well, so there is that option as well as doing year abroad. Yeah.
Great. No, I think I would echo that, Alana. It's well worth coming and taking a look. Even though I grew up in Kent, I'd not been to the campus till I came down for my job interview, basically, from London, and it was such a nice, refreshing change to get out in green spaces. It's a lovely environment to study and to work. Yeah.
Yeah, absolutely. It was coming down onto the campus that I think definitely made it for me, and that, like, yeah, you can't beat being able to go onto a campus, but unfortunately it's different circumstances. So yeah, if you're able to, it would be great, but do the best that you can. And that I'm also on UniBuddy as well, so if you've got any questions that you do want to ask on here, then you can go through the UniBuddy page and you can ask me directly, so no one else will see that question or comment or anything. That will just be a private message to me through the app.
Great. Right, well, thanks for joining us. Hope you all enjoyed it, and yeah, good luck anyway with whatever you decide to do next.
Yeah, absolutely. We hope you choose us, but you go where you need to go and where it suits you. So
Absolutely.
Yeah, and we've got a few thank yous as well.
Great, always nice. Okay, thanks very much, Alana. I'm going to log off now, and yeah, have a good evening.
All right, bye.
Thank you. Bye-bye.